a microbial world. Today we'll be talking about Winogradsky Project.
In ecology, succession is the progressive change in plants and animals' lives of a region. In order for these organisms to advance, microbes or elements continuously create or aid the growth of other organisms, completing the circle of life. Within ecological succession, there is microbial succession, which is the selection and development of sequential microbial populations in natural or disturbed systems. Sergius Winogradsky and William Burdentrick were curious about this, established a study in which prokaryotes recycled all the mineral elements necessary for life support through the use of the Winogradsky column. Using the Winogradsky column, they could observe how different microorganisms perform independent roles, how activities of one organism enable another to grow, and how columns are complete, self-contained recycling systems.
Hey guys, so in order to make a Winogradsky column, you have to have pond mud, and then you have to filter it so that there's no large particles or other things that you don't want. And then you have to take newspaper or like what we use, paper towels, and you have to put it so that there's 1 inch all the way at the end after you shred it.
After you compact your paper towel, what you do is you mix the mud with 10 grams of calcium sulfate and 10 grams of calcium carbonate, and you, with the glass rod again, you compact the soil as much as possible, so that there are 3 inches left on the top, which will be filled with water afterwards.
Okay, so after you do what Alvin just told you, you have to take plastic wrap and you have to put it on top of the on top of the tube. And then after you do that, you take aluminum foil and you put it like all around the tube. So after you take off the aluminum foil off the Winogradsky column, you have to put it by light source. And the light source should be 12 to 20 inches away from the Winogradsky column, and you should have a 60 watt bulb next to it or as the light source. During this incubation time, there will be various colors that are going to come up because of the photosynthetic bacteria. Thank you for watching how to make a Winogradsky column, and try it yourself. Where's Alvin and Deepa?
I don't know, but I know Deepa's inside.
Okay, cool, let's go. Nothing, I just figured out that there's anaerobic sediments at the bottom of the column. What does that mean?
The cellulose in the Winogradsky column depletes both the oxygen in both the residue and in the water.
Good job. Maybe you should learn something from this. Don't you remember learning in the prize class when you were talking to Edward that there's anaerobic bacteria that's going to grow in here and it's going to produce hydrogen sulfide?
I didn't get what you were trying to say. Can you please show me in a diagram?
I can.
In the bottom of the column, there is anaerobic sediment. This is because the cellulose depletes the oxygen in the residue and in the water. Anaerobic bacteria will then grow and produce hydrogen sulfide. In this case, Desulfovibrio and Clostridium are predominantly the bacteria. Clostridium breaks down the cellulose into glucose, which it ferments to produce a range of simple organic compounds such as ethanol, acetic acid, succinic acid, acetone, butanol, etc. Desulfovibrio reduces sulfate into large amounts of hydrogen sulfide, which when reacting with any iron in the sediment produces black ferrous sulfide. The hydrogen sulfide travels up the column and is used by other organisms. Some of the sulfide might then be oxidized into aerobic surface layers by autotrophic sulfur oxidizing bacteria, such as the chemoautotroph Theobacillus, and regenerate some sulfide. The diffusion also enables anaerobic photosynthetic bacteria to grow, resulting in 2 bands, one of green sulfur bacteria and the other of purple sulfur bacteria. By using hydrogen sulfate, carbon dioxide, and energy from light reactions, they are able to produce their cellular materials. The green sulfur bacteria, in this case presumably chlorobrium, have small cells that typically deposit sulfur externally. The sulfur it produces returns to a sediment where Desulfovibrio can recycle it. This is a part of the sulfur cycle. The purple sulfur bacteria have large cells and deposit sulfur granules inside their cells. Most of the water portion of the column is bright red due to the large population of purple non-sulfur bacteria, including Rhodospirillum and Rhodomicrobium. These bacteria are photoheterotrophs and use organic acids as their carbon source for cellular synthesis. The organic acids used are the fermentation products of other anaerobic bacteria, but purple non-sulfur bacteria are intolerant of high hydrogen sulfide concentrations, so they occur above the green and purple sulfur. Any hydrogen sulfide can be oxidized by sulfur oxidizing bacteria. Cyanobacteria can also grow and are the only bacteria that have oxygen evolving photosynthesis.
Because Alvin doesn't know what he's doing.
Oh, really, Deepa? Well, you know what? I got you with the bloopers. Come on, the first thing with the bloopers.
Long rod to like make sure that there's not that much air. Oh my god! And after 4 to 7 days, you have to take off the aluminum foil and you have to. Damn it! Guys, where's Alvin? Guys, maybe you should like listen in class except rather than like talking to.
Well, folks, that's it for now. This has been an A-Day introduction. Deuces.